So with that, I want to open up our first session. We have three excellent speakers here today. Our first speaker will be Professor Arnab Pain. And very impressive bio here. I'll try and read through. Professor Pain received his PhD from the University of Cambridge, working on microbial pathogenesis and bacterial pathogens at the Weatherall Institute of Molecular Medicine in Oxford. And while there, he focused on cell biology and virulence-associated phenotypes in the human malaria parasite. He then moved back to Cambridge and joined the pathogen sequencing unit at the Wellcome Trust Sanger Institute. And in 2010, he moved to Saudi Arabia and joined the Biological and Environmental Sciences and Engineering Division at King Abdullah University of Science and Technology. And since 2015, he has had an appointment as a distinguished professor at the International Institute for Zoonosis Control, Hokkaido University in Japan, and also serves as an honorary professor at the Liverpool School of Tropical Medicine. He'll present on genomic surveillance and functional genomics in a cohort of COVID-19 patients from Saudi Arabia. Dr. Pain, welcome.
Thank you. Good morning, everyone. And thanks, John, for the introduction. And before I start, I'd like to thank the UK ICN for inviting me to share some of the work that we have been doing, and also the organizers of this meeting, Tatiana and her colleagues here in this beautiful city of Ljubljana. It is my first time, and I really love to do a little bit of touristic activities on Friday after the end of this meeting. And also, I'd like to thank Zoetis for sponsoring today's workshop. So as you see that I am going to talk to you about some work that we have been doing in Saudi. And I must say majority part of the talk is a result of some collaborations or networking activities under this umbrella of UK ICN. So I thank Julian and all colleagues at the management committee for giving me an opportunity to interact with you, because by training, as you probably have seen, I'm a malaria person. I work on parasitic proteins. So for me, viruses are really a new beast, to be honest with you. Okay, so let me start on that. Okay, so basically, all we have been doing, what many of you also have been doing, but this is related to Saudi, that we have been, and this is, I'm talking about right at the start of the pandemic when there were no vaccines. So during the first and second peak of the second wave, of the disease, we have been systematically collecting samples, nasopharyngeal samples, but instead of VTM, we have been collecting them in trisol to make sure that the RNA are kept and transported in a biologically safe way. And then we have been looking at various aspects of that collected sample. So we are looking at the viral genomes. At the same time, for the same sample, we have been looking at the transcriptome, for example, in nasopharyngeal swabs of those COVID positive individuals. But also, as you know, I just mentioned that we collected the trisol. So the organic phase, we have optimized the protocol to collect the proteins. And then we did a large-scale proteomic study as well. But for the purpose of today's talk, I'm only referring to the genomes and transcriptomes and some functional work that we have been doing as part of ongoing work. So this is literally to summarize the work that I'm going to talk to you about today. So I have actually, at the start of the pandemic, I realized that there were not huge amount of coordinated effort happening on the sequencing front. So I basically formed a consortium. I call it Covidomics KSA, Kingdom of Saudi Arabia. And then I basically looked at the genomes, also the transcriptomes, and also follow up, basically based on what results we found. We basically then following up in the lab with different systems. I will talk to you, refer to in my talk. And also, which I'm not going to talk about today, is we are also looking at the viral RNA changes. So the AP transcriptomic changes in the viral RNA, in infectious RNA molecule, and see how it affects the COVID outcome. Some of the work has already been published. This work, actually, I already spoke last time that UKICN meeting that Walid had very kindly organized. In fact, that prompted me to start collaborations with some of you. You'll see during the course of my talk. And also, we followed up on other things. Some of them are just published. Some of them are still kind of remains unpublished. Okay, so before I start, as I told you, I'm talking on behalf of a large army of people, very dedicated individuals. A lot of them are my collaborators from Saudi Arabia, from different hospitals. I just mentioned a few of them here, particularly Dr. Sharif Hala, my former PhD student. Now he runs his own group in Keynote and National Guard Health Affairs. Then also Taiba University, King Abdullah Aziz University, Anwar and Ahmed and Abdullah. Then several doctors in the Ministry of Health hospitals. They played a critical role in collecting samples during those times when, there were really the situation was quite grim at the time. Then also some other doctors from private hospitals like Suleiman Al Habib Hospital, and also from King Faisal Specialist Hospital, Zumana, helped us quite a lot. At that time, we also, of course, my own group members, they played major role, particularly Tobias, he's a bioinformatician, and then Muhammad, he does a lot of, you'll see a lot of mention of his name during the talk. He's a really talented research scientist with a lot of expertise on molecular biology and cell biology. We also other people who have contributed during the time of large-scale, sequencing and sample collection operations. We also collaborated during the first phase of the study with Eric Waltz and David from Imperial College London. And later on, you'll see as I speak now, some of the work that are happening, particularly with James Stewart and his group in Liverpool. We also did a little bit of work with Giancarlo in LSTM, and also some work has just started with Nicholas Matheson in Cambridge. Of course, when it comes to funding, initially it was funded by KAUST and CAXT and also Ministry of Health. Now it is very generously funded by this new initiative called KAUST Smart Health Initiative. And also I'll show you that we have now received generous funding from the Wellcome Trust to work on the large-scale genome surveillance in our part of the world. So now I want to introduce you to a particular set of co-occurring mutations in the nucleic acid gene. As you know, most of the studies on coronaviruses, particularly COVID, have focused on the spike protein. But during our study, we actually found, and I'll take you through that, what we found, that the nucleic acid gene is really, really important in determining the disease outcome. Now, I just show you a couple of a mutation, which is this KR mutation during the time of, until say November 22. So this particular mutation appeared to come and then got fixed around this time. Now I just took this picture from next trend. So basically this color, if you follow, this is the KR. So now it is completely fixed in population across all this. In fact, this is the latest BA 2.86. It's all there, 100% of the samples. That particular mutation, I'll tell you why this would come, this particularly relevant and why I'm just picking up this mutation. Another very interesting thing that we observed by comparing all those millions of data sets in that this mutation appeared in quite a bit of like I would say it appeared here in alpha, went completely out in beta, then reappeared in gamma, completely out in delta, omicron. So the black one is with KR mutation of all GIS AID depositions. And now, of course, I showed you that 100% of them, of all omicron, they have this KR mutation present. So when we started at the first stage of this pandemic, we basically started sequencing, analyzing the data, putting it in GIS ID to make sure that everybody else in the world get access to the data. What we also did, we actually collected a lot of metadata, like sample, like the patient's status, if they were alive, their ethnicity, and the state of disease, severe disease, mild disease, and so on and so forth. What we found that we actually discovered that this mutation, we did like a global case control kind of study based on mutation and outcome, and we found that this particular mutation, KR mutation in the N gene, was associated with higher viral load in our study population. Of course, we did a lot of work with it, and then what we saw is that that mutation, we wanted to check what is the functional effect of that mutation. And as you know, the nucleocapsid gene is a very important protein for viral life cycle. It involves RNA packaging, transcription, replication, and also it has already been shown in various coronavirus studies, including COVID, that perturbation of the host cell processes by N gene could be related to from interferon production in the host cell life cycle, cell translation machinery, and also the cell cytokinesis. Now, where is this mutation that I'm referring to that we particularly homed in on the N gene is here, which is the LKR region. That's sort of a very important regulatory hub for the molecule. And of course, we kind of thought that that particular from RG to KR mutation may be associated with regulation of RNA binding and also interaction with host factors and also phosphoregulation. So what Mohammed actually, I need to move that somehow, somewhere. Maybe this, okay. So, so Mohammed, then we started off with a lot of functional work to see what are this functional impact. So I have actually already talked about them in details, and that's already published in the Nature From paper. So I'll just take you through briefly what we did without showing the results and just summarizing them. So basically, we look. He expressed these proteins both in N like normal form, Wuhan version of N gene, and with the KR mutation, everything else is identical. Looked at interaction with viral RNA. We also looked at using oligomerization by BS3 cross-linking to see the effect of the mutant N on the Wuhan N. We also looked at interaction with host proteins in the sort of in HEC293 cell models. We also looked at, we did a phosphoproteome analysis of cells transfected with the N and the mutant N. And then finally, host transcriptome in two types of cell lines, HEC293 and also CALU3. And also at the same time, just to prepare us for looking at what happens with either VLPs, so pseudovirus particles, or with the real reverse engineered viruses with specific mutations that we are studying at them. So we just published that, and what we concluded in that paper last year was that, of course, as I have been showing you, but I haven't shown you the data, but just basically we established that this particular mutation is associated in the nucleocapsid gene, associated with higher viral load and severity in COVID patients. We also looked at this N protein as higher RNA binding, so in real samples, real COVID RNA samples from patients. We also had shown that the N gene displays increased interaction with the host and has a higher oligomerization. In order to assemble the virus, the N gene plays a key role. The oligomerization N gene is an important step, and that particular mutant aided in that thing. Also, we looked at, it's a very specific serine 206 phosphorylation site, which is super hyper phosphorylated in the nucleoside mutant N, as opposed to the Wuhan type of N that we had shown with biochemical experiments. And then finally, with the global transcriptome analysis of the transfected cells with mutant and wild type or Wuhan type N showed specific hyperexpression of interferon-related genes. So this is what we at that time, we wrote everything up. Then we thought, okay, let's see what's happening in real patients, COVID patients. And we had access to all the virus. So basically, we knew each and every patient what viral genotypes, with what kind of mutation SNPs they had seen. So it was, for us, we wanted to see that are there particular this mutation, what we are seeing in vitro assays in transfections, are also reflected in real patients. So we then embarked on this project, which is we call RG patients, so patients who had seen, irrespective of what other mutations they had, at least in the N gene, they had the original Wuhan type, and then this is the KR that I'm talking about. And of course, idea was they were basically matched with age, gender, comorbidity, ethnicity as closely as possible. We had like 3 types here: healthy patients who were COVID negative, but came to the hospital for some other thing, and then RG patients, COVID patients, and also. So what we saw, this is the first time that in all those patients, whenever there was presence of this KR, there were many number of genes. I'm talking about global bulk transcriptome here from nasopharyngeal samples, and you can see the much number of genes are high expressed. And then when you do a PCA, you can see the green ones, which are KR patients, sort of tend to cluster together. And what we see is particularly perturbed transcriptome in the patients who had seen the KR version of the virus. Now, this is just basically showing you an impression of this is the KR patients, this is the RG patients, and these are top 50, for example, upregulated genes, if you like. And you can see that there is definitely a clear visual difference and statistically significant difference of this lot here as opposed to the other categories. Now, if you look at the gene ontology enrichment and all those bioinformatic measures, we clearly saw that there have been some major upregulation, for example, of interferon inflammatory response, viral defense response, and other ISG and cytokine-related genes upregulated in the KR patients. So we also wanted to look at the validity of our data. So we are using another data published in Cell Host and Microbe on bulk samples. So bronchiolar, alveolar, lavage samples, fluid samples, and looked at they had this list of genes that were particularly upregulated in severe COVID patients. And I just show you a visual impression here. Again, the pro-inflammatory cytokines, for example, displayed a particular higher regulation patterns of expression in those patients. And also, we looked at another Cell paper, quite a seminal paper on where they talked about the entire repertoire of interferon stimulated genes, ISGs. So we took the list, and then we looked at our data, so in our samples, and we see again a significant overlap in our KR patient samples. So then what we did, we had the bulk transcriptome from all our patient data, and then we used a machine learning based approach that was already very highly used and highly cited, developed by Stanford, where bulk data can be then segregated into single cell data sets based on transcriptome profiles. So when we basically applied our data, we see that actually, and this is you can see this is the KR patients, and that particular color, if you follow from here, these are actually neutrophils. Of course, based on single cell as deduction, if you like, of cell types in our data sets. And this is what it looked like in RG and in healthy. Now, so seeing that this is individual data sets, what you saw before was like a average. So you actually see that these are all KR patients here. You can see here, and they have much more neutrophils there in their samples. And of course, there were many papers. I just show you one of them, that neutrophil to lymphocyte ratios is shown to be a major predictor for critical illness in coronavirus diseases. And what we saw exactly very similar pattern, that we see much more neutrophil. And this is the ratio here, for example, where based on our global transcriptome data, that we have the KR patients seem to have much more NLR ratio in our data sets. So at that point, we thought, okay, let's also go further deeper into mechanistic and see that, okay, whatever we see, it's not just NR, because these are patient samples. You have various patient genotypes, various viral genotypes, except the KR. So we just differentiated between KR and non-KR. So let's do much cleaner experiments to prepare some virus-like particles using this particular system. So where you can mutate your wish all these various structural genes, and then you can actually, which is associated with luciferase promoter here, and then you can transfect these cell lines, and then you can either do transcriptome proteome or you can do a luciferase assay to see the infectivity. So what we chose 4 types of virus. One is completely, I'll show you later, yeah, here. Wild type, so everything is wild type. Of Wuhan type S and N, this is the KR, so everything is except just a KR mutation in the N. This is D614G, which is already very well-known spike mutation, as you know, widely reported. So that, and then combination of these two, D614G in the spike and KR in the N. So when we transfected this, we see this result, which was very encouraging, that the moment you have D614G KR, you have much more luciferase expression, of course. This is compared to wild type. So we knew that it is definitely, even in an absolutely clean background, genetic background, we see the results. And then when you do the transcriptome again, we see that if you follow that KR here, for example, the same genes are hyper basically expressed or overexpressed, if you like, during in our study of the transfected cells. And also the same pathways, when you look at from using the data, we look at the Go analysis, we see the same. So what we did, because we had the cells, we also wanted to see that at proteome level, do we see the same evidence or not? Exactly what we have done, same VLPs, and these are so anywhere we have the KR with this, or just KR on its own, these are the green and the blue here, you see that their pattern of protein expression is also quite similar. And again, the same pathways appear to be hyperregulated or upregulated in these patients, in these samples. So, and this is just to show that our proteome and transcriptome from the same cell lines significantly overlap and would basically show the same biological phenomena here. So what we also did, we used this cytokine storm kit for the same cells to look at the expression of these various markers, which are basically known to be markers for cytokine storm. And we see at least in 4 out of 6 different molecules that we tested here, we see significant difference in our samples. So this is the summary that, of course, just published in Genome Medicine, where we have shown the effect of this structural effect of this mutation in engine and how it is upregulating various pathways, including inflammatory response and other. And this is perhaps one of the, I would say, one of the examples where how just a couple of mutation in the virus critically determining the host response. And actually, we also have a similar work, but on large-scale proteomes across all these individuals, but it's about 300 of them under review at the moment in a journal. But then comes to something that we haven't yet published. It's work in progress, and I must say a lot of that work actually started off after the last meeting, UK-ICN meeting. So I owe all these things to UK-ICN network for allowing me to network with real experts in the field. So we actually now started working with animal models and also cell culture. So various peoples are critically involved here. So basically, again, we have got all these reagents from UTMB, where we can now actually reverse engineer SARS-CoV-2 as the way we wish. So basically, that allows us, so we basically created exactly the same virus, but real virus, infectious viral particle, and full-length RNA were prepared by this N and N mutant, again done by Muhammad. And this is, again, the protocols were published here by whoever the group produced this. But these reagents we brought to KAUST and made all these different types of real viral full-length RNA. Then they were transfected in Giancarlo's lab in LSTM, and they all 4 out of 4 created infectious virus particles. And then we sequenced them. We saw exactly the way it should be. So their genotypes were exactly as the way we wish. And then James played a key role in we have been collaborating, and we actually have done the first sets of mouse experiments already. I'll show you one set of example results now. And also we are now just started collaborations with Nicholas Matheson in Cambridge, in Edinburgh, where we are going to do some in vitro experiments with some reporter viral construct that they have generated in Cambridge to see the effect. So basically, the idea is to look into mechanistic aspect of this mutation to understand how, how we see what we see at the phenotype level. So this work is really kind of just few days results available, just few days before. So, and I must say that, I must want to thank Catherine, Parul, Adam, Eleanor, and Daniele, and also Anya to kind of take part in this analysis and work. So basically, as you know, we have got this designer viruses, reverse engineer viruses of our choice. So our genotype, and then they had infected this. And of course, we have the first weight loss data analysis of all these 4 types of viral. You can see that it's actually the moment you have the D614G and KR combination, spike and N, you have got a significant pathology. We already know that in some other work that's happening at the moment, and they actually this particular mice had to be sacrificed because they were really under stress. And also the first set of lung viral load, you can see this clearly sort of what we saw in a genome study, in a genome surveillance study, we now actually see in a real experiments in a very clinical genetic background that we actually see when D614GKR, you have a much higher viral load than just Wuhan, everything, and then D614G here, KR on its own here, and then together it's even higher. We are actually also started getting a lot of work on histopathology data, actually looking really, really cool, but I think it's too early at the moment to kind of immediately describe, but we are kind of making biological sense of what it means. So just last part of my talk, how much time I have? Okay, 2 minutes, that's fine. So basically, as you know, I started off with genome surveillance, and this is what I have been doing a lot of part of my recent career, if you like. So when I looked at the viral genome GIS AID submissions, it came loud and clear to me that we, based in the Middle East or also in North African region, are doing really, really poorly. And I tell you what, this data comes from just 2 days old. See what happens. We basically have contributed only 1.3% of the global GIS AID submissions. When I say we, I mean all these together countries in the Middle East and North Africa. And of course, KSA Kingdom only done 0.33. And I'm saying not about how many have been sequenced, how many have been made publicly available, and that is critical for this kind of analysis, because data should be made globally under this kind of pandemic situation. So what I did was, I then thought, okay, this is a major scope and perhaps need to start a consortium. So I basically followed the UK COVID genomics, if you like, model, and then started calling something COVID genomics MENA, which I contacted many collaborators across this Middle East, North Africa, based on all these different 7 countries, actually, if I consider Saudi as one. And I am actually serving as the lead PI on this, and basically Wellcome and Foreign FCO has funded us generously to go for this large-scale genome surveillance for COVID. And the idea is really to not only use COVID as an example, but to prepare ourselves for future pandemics or other emerging infections, including viruses. So saying that, let me just conclude. I have shown you omics-driven technologies and how big data analytics to track changes in the virus as in a pandemic situation, and we applied it in Saudi Arabia. We showed particular mutation, how it in the nucleic acid gene and its importance in determining pathology and also outcome of COVID, not only in real COVID patients, but also in experimental infections. I also showed you that what our plans are on our ongoing experiments on looking into mechanistic aspects. I mean, at the moment, I think we are very close to determining how this particular protein played a key role during the first phase of pandemic in I would say, instigating cytokine storm in patients. And as you remember, a lot of people actually died of cytokine storms in the first phase when there was no vaccine and other immediate prophylactic measures available. And finally, I have not been able to show any data at the moment. Hopes next time I get a chance to speak to you about what we have done as part of this COVID Genomics Meta Consortium. And just before I end, I want to show that while we are doing, other people are also independently have discovered the same mutation and have worked on it. Some came around the same time we published. Some came after we published. But the important thing is, I feel very kind of relieved that whatever we saw also been independently been verified in other labs about the importance of this mutation. And finally, I again want to thank the organizers, the sponsors, and I'll be very happy to answer your questions. Thank you.
Thank you. Did you ever go back to see how widespread the KR mutation is across herbaceviruses?
Across the?
Herbaceviruses, so all your SARS-like viruses in animals and humans.
We did look at least in SARS-CoV, and we did not see any KR mutation when we last looked at it. But not very recently, we have, because it's kind of an ongoing something, but we haven't found this. What really intrigued us was, what was it driving? It was coming back, alpha it was, beta went away, gamma, delta it came back. So it's kind of a very puzzling, but what we see that we have seen some unpublished data where people have tried to sort of culture it for a long time, like as Yuhan in animal models, of course. And this mutation appears. And once it appears, it stays because it's giving better fitness. And all these papers, our own and others, show that it gives the virus higher fitness to survive. But of course, what you're seeing is just this mutation, but there are others. So basically, one of our future studies is, okay, this is important, we have already established, but what are the other, because Omicron on its own right is much milder, right? And it has these two mutations. So there are other mutations which are doing probably a balancing act. So this is something we want to study in future, and we are sort of just in the process of writing a big grant on determining what are the other factors that are acting as a balancing factor. Thank you.